So hi everyone. So I'd like to tell you about a way to approach aging and health span from point of view of physics. My group at Weizmann combines mathematical modeling and big data analysis and some experiments with Valerie, for example. And of course, the idea is that instead of treating aging-related diseases one by one, we can target aging itself and reduce the risk of diseases. And we know that also in the field of aging, there are many theories of aging, and there's this picture called the hallmarks of aging that, in my view, is like a peace pipe. It's like if you work on any one of these aspects, you're one of us working about aging. And problem is that they're interconnected and very difficult to make sense of. So many changes happen during aging. Are there ones that happen before and then later, or is it all happening in the same time? And since I come from physics, I have this disability that I need to pretend that there's some simplicity in the world, at least some angle where things look simple. And so that's what's driving our approach, this naive belief that there's simplicity, that there are drivers of aging that you can address that will help cut through the complexity without ignoring the complexity. And the way we did that is using mathematics, and I'm not going to tell you the equation, I'm just going to give you a metaphor. So first of all, how do we get to a model of aging? So we start from a lot of patterns that happen in aging in many, many organisms. For example, the risk of death rises exponentially with age. The risk of diseases rises exponentially with age and then goes down. If you make a change in midlife that extends life, your risk of death almost immediately goes to the better risk. So there's a slow time scale, a fast time scale, and there's also randomness. So if you take genetically identical mice in the same conditions, they die at different times, just like unrelated mice. So there's also random aspects to aging. And put that all together, you get an equation that has long time scale, a short time scale, and noise. And in physics, we really know how to handle questions like that. And the way we got to it was by looking at, with Valerie, at senescent cells as a function of time in mice and seeing how they go up and down, and asking what equation describes that process of production and removal of damage. And we got from that data to an equation that makes more and more sense to us as time goes by. And the way it works is this. So there's a garbage. The garbage, I'm going to tell you what it is. We think in human beings it's going to be senescent and damaged cells. And the reason it rises with age, in this model, there's like damage-producing units, like factories that make the garbage. And there are trucks that remove the garbage. This is senescent cells that Valerie talked about. Immune cells, like NK cells. And that's fine. This village is working, and there's some amount of garbage. The problem is that with age, you make more and more of these factories. And we didn't evolve to be old, like Chaim said. So for some reason, the number of trucks doesn't rise with age. And now we have a problem. Tons of garbage is being made. The trucks are overloaded. Garbage piles up in the street. If there's a truck strike, like the immune system is fighting an infection or something like that, and garbage goes through the roof, and soon enough it becomes incompatible with life. Those are the elements of this mathematical model. Something that irremovable kind of damage-producing units that rise with age make a damage form that's deadly and saturates its own removal processes. And this model is agnostic to what this garbage is, so you can use it to understand yeast, mice, worms, etc. Each time it's a different kind of damage. I think in mammals it will be something like cellular senescence. And we can use this model to actually find the biochemical pathways. And that's very exciting, because if you know the biochemical pathways, you can think of interventions, just like Chaim and Valerie said. And the way we do that is the model allows you to use a graph that's used very commonly in aging research, and we saw some of them, which is a survival curve. So you take some, let's say, mice, and you ask how many of them survive to a given age, and then you give them an intervention, and they survive more. And usually we look at the maximum survival or the median. Our model helps us also interpret things by the shape of the curve. So does this intervention move both maximal and median lifespan, or does it change the shape of the curve or not? And that gives us information whether it affects the trucks or the factories by the shape of the curve. Changing the factories makes you live longer, but the same shape, and changing the trucks makes you live longer, but a steeper shape, for example. And with that tool, we can start unraveling this network and find out candidates for the drivers of aging. So most information we have, and that's very useful, is on yeast that makes buds. And for some reason, the mother cell divides, and then dies. And the genes involved in that are very similar to mammalian genes, like for instance, the acetylation genes that Chaim works on were discovered first in this budding yeast, etc. So another thing about yeast is we have thousands of mutations and we have data about their survival curves. So we analyzed 30,000, basically it's a genetic screen for which mutations make the yeast not only live longer, but also change the shape of the survival curve, make them die more at the same time or actually more randomly. So this is like a genetic screen that powered by this mathematical model. These are the longevity and steepness of all those different mutations, and you can see that there's a lot of information there. Only 10% of them change the shape of the curve, and this genetic screen gives you genes that are very informative. They cluster into specific biological pathways that you can interpret, and what we find there are basically two kinds of garbage, two modes of dying. The first one has to do with proteins. The factories are these mistranslated proteins that become aggregates, and they're cleared by autophagy, many things, but the bottleneck that gets clogged up and kills the cells is a weird way to get rid of aggregates inside the mitochondria. So we find all these mitochondria quality control as trucks, and then the mitochondria die and the yeast die. And it's known that yeast can die in two different ways, either mitochondria with small buds or DNA-based damage with elongated buds. And indeed, in our way of looking at things, we find that there's two kind of pathways, at least. Or aging is like an onion. You get rid of the worst kind of damage, you'll have the next one. Then you get rid of that one, you'll have the next one. The next one is a DNA-based one, where histone acetylation opens up telomeric regions, causing all kind of secondary structures that collide with the replication fork, and the trucks are all kind of resolvases of that, and that's what kills you. And many of these genes are also, when mutated in human beings, make people age much faster. That's called progeria, like the Werner and Bloom helicases, etc. So we think, so by this mathematical model that we derive based on patterns of aging, we can have a new way to interpret what mutants do, whether they affect production or removal, and come up with candidates for pathways of aging, or at least put a lot of the hallmarks in a certain order. In mice, when we do the same thing with deletion mutations, we find that what happens in yeast also happens in mice on the cellular level, but mice also have immune systems, etc. So those damaged cells become senescent, and the immune system are the trucks, and the rise in in senescent cells, like Varley says, starts a lot of age-related diseases, and that's why targeting those senescent cells is a good place to be. So it's like cellular pathways, like in yeast, two different pathways. We think one is for non-dividing cells, that's the mitochondria one, and one for dividing cells, that's the DNA one, cause cellular senescence, which leads to a lot of tissue problems. Valerie also mentioned this idea that we need to compress the sick span. And it turns out that right now we don't know which drugs, in mice, for example, make the sick span. So you can stretch life maybe by, let's say, 20%. It'll be sad if you also change the time that you're sick by the same amount. But we don't know which drugs do that or not. This model gives a prediction that if you change the rate of damage production, you stretch time like a rubber band, and you make people not only live longer, but also sicker. I mean, at each age you're healthier, but the sick span is proportionally stretched, whereas things that make the survival curve steeper should shorten the sick span, and that indeed is found in worms and, I don't know why this is not working so well. You only see the half of the data, but in the steeper the intervention in worms and flies, the longer the more compressed their sick span. And in mice, there are a few interventions that make survival curves steeper, and they are very interesting because they all bear on, or many of them bear on vasculature. So these are things like Miriam, who's here, work on improving vasculature, makes mice live longer, but also die more at the same time. Senolytic drugs do that, ketogenic diet, and all kind of diabetic drugs like acarbose and things like that improve vasculature, which we think is important in mice and even more important in human beings. So why does vasculature make you steeper? We think the vasculature, part of the reason is they're the roads for the trucks, the immune system, to get in and remove the garbage, the senescent cells. So improving vasculature has many positive effects, but it, we think is likely to compress sick span. So it's a way to prioritize interventions. This model can also help you understand combinations. So if you combine a drug that works on production and another drug that works on removal, like rapamycin and acarbose, you can get additive effects because they work on different aspects of aging, and that works very well according to theory. So just to summarize, we like to pretend that there's a direction in which complex systems look simple. With this idea, we try to build a minimal model for damage production and removal. We find that aging is like an onion. There's one level of damage. Then if you clear it, you see the next one and probably next and next. It gives us tools to find out what interventions do. Do they affect production or removal of damage or noise? And therefore, give us tools also to prioritize interventions and combinations, whether they compress the sick span or not. And the goal of every theory, in my opinion, is to spark new experiments, and this has been very fruitful with researchers like Valerie to test these ideas. Yeah, it's just thrilling to work on this puzzle called aging. So I'll stop here and be happy if there's any questions.
Questions? Please. Did you try to correlate your model with some of the most widespread symptoms of aging, like insulin resistance, which is correlated with so many diseases, or other types, high blood pressure? Did you try to correlate?
Yeah. So the question is, did we try to correlate our models with physiological measurements that change with aging, like insulin resistance that rises with aging, blood pressure? Yeah. So we do. We have access to Clalit health records, and we looked at all 110 blood tests as a function of age. It's incredible that you find already at age 20 a rise in things like blood urea nitrogen. So aging starts very earlier than you expect. And this model also tells you what to look for. So in order to look for drivers of aging, you need to look for something very special. So many things with age become more different, right? You have an 80-year-old that can sit in an aging conference and be very concentrated, and another one that's in a wheelchair. And very, very different. But the drivers of aging that are upstream rise with age but become more similar. All aged people have them. So something like glucose, some people have diabetes, some don't. They become more different with age. But things like inflammation become actually more similar in relative terms, and we think they're upstream. So inflammation drives insulin resistance, for example. So mathematical models like this can help you find a needle in a haystack by looking at the statistical properties that you're interested in. And this combination of big data and mathematical models, I think, is very powerful if you want to ask for a mechanism. Did I answer your question? Okay, yeah.
Any more questions? Please.
Do you think that you can apply this type of analysis in an organ-specific way to analyze the way cells in the tissue
Yeah. So the question is, if we can apply this in an organ-specific way, because you also have survival curves of cells of different types in each organ. Did I get it? Yeah. So one thing, of course, we're working with Valerie to discover the markers of senescence in each particular cell type in each particular organ, and they're very different. And we also apply this approach to disease-free survival curve. So if you think about Alzheimer, disease-free survival curve is not exactly It's also looks like this, and then you can look at different mutations, TREM2, APOE4, etc., and ask, does it change the shape of the survival curve? And for that, we can try to put together a pathway for the disease process, so we can get to glia and to oligodendrocytes and neurons and what's happening in there, and also, when you look at single cells, like in yeast or bacteria, you definitely can apply this model, because that's where we did. So in all those levels, I think it's useful. Thank you very much.